Welcome to the Introduction to Space Biology Data. The objectives of this lesson are to identify model organisms used for space biology research, to describe experiments for space biology research, to define data types and formats for space biology experiments, to find data types and formats for machine learning models, and finally to identify statistically significantly different data distributions. In this first section, we will identify model organisms which are used for space biology research. As we've already discussed, there are several ways in which astronaut health is negatively impacted by spaceflight. For example, here on Earth, it's gravity which tells us up from down. But in space, in the absence of gravity, there is no up and down, and the body and the brain get disoriented. Also possibly due to the absence of gravity, the lungs show a decreased capacity for air, causing some extra strain in respiration. Without gravity, the musculoskeletal system atrophies. Astronaut eyes often develop cataracts, cotton spots, and even change shape as a result of spaceflight, all of which are detrimental to astronaut vision. Radiation is known to cause genetic mutations, which in turn can develop to cancer. All of these risk factors for astronaut health become risk factors for missions in space. In order to conduct space biology research, we need data. Data are gathered from observations in ground experiments as well as spaceflight experiments to model the impact of spaceflight on the health of an organism. These models need to be built, tested, and validated, and all that requires data. Unfortunately, experimental data in space is extremely limited, and data from astronauts is even further limited because it's usually so sensitive that cannot be shared broadly to the scientific community. So, in the absence of experimental data on humans, we turn to other organisms as model organisms. One mammal which is relatively close to humans is the mouse, and most of the data we will be looking at in this course is gathered from experiments with mice. Other organisms, including fruit flies, worms, plants, and microbes, are not evolutionarily close or physiologically similar to humans, but are still informative in their own right as to the impact of spaceflight on living things. In this section, we describe how experiments are conducted in space biology research. The best space biology experiments are conducted in space, but they are also the most expensive. The International Space Station has been in low Earth orbit since 1998 and conducted thousands of space biology experiments using all those model organisms we discussed earlier. But each experiment only has a few samples because there's such a high cost of transporting them and conducting the experiments and returning those organisms back to Earth. To compensate for that, space biology researchers simulate the effects of spaceflight on Earth. One simulation they do is to simulate the effect of microgravity by spinning organisms in a centrifuge. Controlled space biology experiments are conducted with the experimental group in space and the control group here on Earth. Their conditions are matched identically, including temperature, pressure, humidity, food, water, light, size and type of cage, etc., so as to precisely isolate the effect of spaceflight on health. Molecular experiments are conducted to determine genomic, transcriptomic, and proteomic level differences between spaceflight and ground control samples. For example, a genome-wide association study is an observational study of a genome-wide set of genetic variants in different individuals to identify variants which are associated with a trait. RNA sequencing is a technique that uses next-generation sequencing to identify and quantify RNA molecules in a biological sample, providing a snapshot of the genes being expressed in that tissue at that point in time. Other molecular experiments not shown in this slide include mass spectrometry, which uses ion beams to identify and quantify protein molecules in a biological sample, providing a snapshot of the proteins transcribed in that tissue at that point in time. RNA sequencing has two primary components: a wet lab component where chemical reactions are used to extract, purify, amplify, and sequence RNA fragments, and a dry lab component where those RNA fragments are quantified and analyzed. This specific type of experiment tells us which genes are being differentially expressed between spaceflight and ground control samples. These differences in gene expression then tell us which biological pathways are being turned on or turned off in response to the conditions in space. As opposed to molecular experiments, which are used to quantify genomic, transcriptomic, and proteomic differences between experimental groups at the molecular level, phenotype experiments measure the response of the organism at a cellular, system, or even organism-wide level. For example, behavioral experiments may measure the experimental effect on model organism behavior, such as maze performance, gait characteristics, and climbing. Microcomputed tomography is a non-destructive imaging technique which uses X-rays to provide high-resolution, three-dimensional images of bone structure. Not shown in the slide, immunostaining methods use dyes, enzymes, and other reagents to detect the presence or absence of a particular protein, characterize its distribution across tissue, or analyze its subcellular localization. Staining tissue samples and studying the stains under magnification can help scientists quantify the presence or absence of certain proteins. For example, oil red O is one such staining technique that stains lipid molecules red. Looking carefully at the two images, it seems the one on the right has more than the one on the left, suggesting there's a higher lipid density in that tissue. However, images such as this need to be quantified, exactly what percentage of the slide is red, in order to be used in research. In this section, we define data types and formats for space biology experiments. As a reminder from earlier in this presentation, in the dry lab, scientists transform raw data into biological insights. Those biological insights in space biology research characterize the impact of spaceflight on the health of an organism. In the case of RNA-seq molecular experiments, the RNA transcripts are sequenced nucleotide by nucleotide. In the example in this slide, the RNA sequence is reverse transcribed from RNA back to DNA so scientists can map these sequences to the organism's genome. In this way, we can quantify how much of each gene is being expressed in this organism's tissue at this point in time, providing a snapshot of the response of the organism to its space environment. This is the raw data that scientists in the dry lab start with. After the RNA transcripts have been sequenced and mapped to a genome, scientists know which genes may be over and underexpressed between spaceflight and ground control samples. In this slide, ground control sample 9's gene ENS MUSG142 has 357 transcripts. Scientists use these prepared data to compare ground control gene expression to spaceflight gene expression. Just as with RNA-seq, which quantifies gene expression, phenotype experiments, which involve staining and imaging techniques such as oil red O, also quantify the presence of some molecular protein. And just like RNA-seq needs to get mapped to a genome in order to get quantified, microscopy images need to be quantified somehow as well. In the case of oil red O, it gets quantified by using software programs that count the number of red pixels in the image and then representing that as a percentage of the entire image pixels. In this example, the slides on the left has 12.02 red pixels and the slide on the right has 17.43% red pixels, both indicating the amount of lipid proteins in that tissue at that point in time. And just like with RNA-seq, we can then put those values into a tabular file, such as what is shown in this graphic. The last column of the table represents the oil red O percentage, which is 17.43 for the spaceflight sample number 5. In this section, we define data types and formats for machine learning models. Two-dimensional image data can either be stored in grayscale or in red, green, blue, or RGB scale. The image is represented as an array of pixels, each of which represents a color or grayscale value for that pixel. Image processing tools such as OpenCV pillow, and image J know how to interpret those files in order to analyze and manipulate the images they represent. Tabular data, as opposed to imaging data, contains strings, integers, decimals, Boolean values, etc., and are arranged in an arbitrary order of columns and rows. A column in a table is a vertical cross-section, and a row in a table represents a horizontal cross-section. Typically, rows represent samples, one row per sample, and columns represent observations, data and metadata, about that sample. A cell in a table is the intersection of a row and a column. For example, in the table in the slide, sample F13's retina measurement is 0.089. Tables can contain both numeric type data and non-numeric type data. For example, the column named spaceflight has values either ground or flight. Note that if there are only two possible values for a column, it could be represented in numeric form, for example, 0 and 1. The oral positivity percentage column has numeric values. These too could be converted to classes such as low, medium, and high, or just low and high, or again, numbers. Numeric values can further be subdivided as integers, which are whole numbers, and floating point numbers, or just floats. In this example, the counts for the gene ENS-MUSG1 are obviously integers, but the values can be normalized across the table to account for discrepancies such as batch effects and different sequencing depths. We'll cover these topics and normalization in more detail in a later lesson. In this last section, we identify significantly different data distributions. One way space biologists can determine if there's a difference in response to ground control and spaceflight is to examine the means of the observations. If those means are very far apart, then we can argue that there's a significant difference in response. But statistically speaking, we'll need to be more rigorous than just waving our hands. In this slide example, the flight samples F15 through F20 have a mean value of total area of 0.252, whereas the ground samples GC15 through GC19 have a mean total area of 0.258. How significant is this difference? It's hard to tell by just looking at the numbers. It's easier to compare the distributions of the data graphically, but not always. Box plots are simple ways to graphically show a distribution of the data. The orange line inside the box represents the arithmetic mean. The top of the box is the 75th percentile, and the bottom of the box is the 25th percentile. The two box plots look similar, but the scale of the y-axis is too small to really know. In this example, the means, the orange lines, look relatively far apart, which would imply that the means are significantly different between the two groups. However, just using our eyes is not a very scientifically rigorous way to quantify that difference. There is a method to compare the means of two distributions called the t-test, and it calculates a statistical value t shown in the slide. x1 bar and x2 bar are the means of the first and second distributions respectively. S1 and S2 are the standard deviations, respectively, and N1 and N2 are the sizes of those distributions, respectively. Plugging those values into this formula gives a float value for t. We then look up this value of t in a table to get the associated p-value. If that p-value is less than 0.05, then we can say those means are considered to be statistically significantly different. Of course, as programmers, we don't want to do any calculations by hand, but would rather automate everything. Instead, for this particular statistic, we can provide the list of values for the flight samples and the list of values for the ground control samples to a function. In this case, it's called t-test_ind, and then examine the p-value associated with that statistic. P-values range from 0 to 1, and the closer to 0 means the more significant the difference. If the p-value is less than 0.05, the means are significantly different. In this example, in this slide, the p-value is 0.88406, so the means are not statistically significantly different.